Dear students, in this part of the video, we will be talking about the cellulose digestion among invertebrates. So in the previous two sections of this particular topic, we have discussed various ruminants, including the cattle. We have discussed the stomach of camel, and we have discussed the stomach of a sheep. So now, after the ruminants, now we are to the invertebrates, that how they are digesting the cellulose which they are eating. So here I want to quote one example of termites. So as we have seen termites in our homes very well, and those termites, what they are eating is the wood. So whatever kind of the wood material they are getting, they are eating it. So termites are known as the enemies of wood, and wood is like full of cellulose. Now how those termites will be digesting this much quantity of cellulose in their small digestive systems? So this is a very important question, and this is a very important topic. So you may get a full-fledged question on cellulose digestion among invertebrates. So now we will be taking some of the examples of invertebrates which are known for their cellulose digestion. So let us start with it. So the very first example that I am quoting over here is that the cellulases, they have been found in the digestive extracts of earthworms and some of the snails also. Here we have quoted two examples. One is of Helix and second is of Limnia. So both are snails, both are mollusks, and earthworms are annelids. So you must have heard of Limnia in the case of Fasciula hepatica. So this is a secondary host for Fasciula hepatica. So here the cellulases, they have been found in the digestive extracts of these animals, and the wood boring bivalve, the pterido. So this is a very important example of cellulose digesting invertebrates. So here I am quoting four major examples. The wood contains various carbohydrates of different types. It is containing cellulose, hemicellulose, lignin, starch, sugars, and the wood-dwelling animals which are living in the woods, they utilize these different compounds. So they are taking up such kind of materials as their food. So one thing is very prominent from this line, that these wood-dwelling animals which are taking wood as their food, they are not only taking in the cellulose, but there are other materials also that they are eating. They may contain carbohydrates, it may contain hemicellulose, lignin, starch, and sugars. So carbohydrates are as well as sugars. Now in the digestive tract of pterido, the wood, the eaten wood, it loses 80% of its cellulose and 15 to 50% of its hemicellulose. So that means that the pterido is able to digest 80% of the cellulose in the ingested food or the ingested cellulose. 20% may be given out as undigested cellulose from the digestive tract, but 80% of cellulose is digested in pterido. And if we take the hemicellulose, 50%, up to 50% of hemicellulose may be digested in the digestive tract of pterido. So this is a very good percentage of the cellulose that has been digested in the digestive tract. So pterido is a very good cellulose digesting animal. So coming back to the very first line, that cellulases, they have been found in the digestive extracts of these animals. Now we have to see that if these animals, they are producing their own cellulase enzyme, or they are having some microorganisms in their intestine which are producing this enzyme cellulase. So this is a very important thing to know, that if these animals are making their own cellulases, their body is making cellulase, or they are having some symbionts or symbiotic microorganisms which are making cellulase for them, as we have seen in the case of ruminants. So that was a case of mutualism. So that is a mutualistic cooperation that was going on in ruminants. So we have to study it in vertebrates also. Now the very first example that I am quoting over here is of shrimps. So we are not taking or seeing or visualizing these shrimps as cellulose digesting animals, but shrimps, they are well known for their cellulose digestion with the help of symbiotic bacteria. So here it is very clear that shrimps, they are not producing their own cellulase enzyme, but they are having symbiotic bacteria. So this is the thing what we were discussing in the very previous slide, that if the animal is having its own cellulase or it is having the symbiotic bacteria. So in the shrimps, we have got the second case, that they have got symbiotic bacteria in their intestines. But when shrimps were exposed to antimicrobial substance, they lost their cellulose digestion quality. So if these shrimps, they were exposed to some antimicrobial substance, if they are given orally or injected with antimicrobial substances or chemicals, so what these antimicrobial compounds will do, that they will wash away, they will be giving a negative factor to these symbiotic bacteria, and the quality of the cellulose digestion in the shrimps will be reduced. So this is very obvious. If we are taking antibiotics whenever we have got a fever, so we take some antibiotics, maybe amoxicillin or any diverse broad spectrum antibiotic. So what that antibiotic will do is that that antibiotic will wash away our intestinal bacterial fauna. So here also the shrimps, they are devoid of their cellulose digestion quality. But when these shrimps, they were re-given a dose of symbiotic bacteria, they again started to digest the cellulose. So that was a very good experiment that was done on shrimps. So after giving antibiotic, they lost their cellulose digestion. But when those shrimps which were given the antibiotics, they were re-given a dose of symbiotic bacteria, so they again started to digest the cellulose. So that means the intestine of the shrimps, it can hold those cellulose digesting bacteria, and the symbiotic bacteria may recover their space inside the intestine of these shrimps. So this shows the role of symbiotic bacteria it should be the role of symbiotic bacteria in digesting the cellulose. So that was the example of shrimps. Next we have some snails which are known for their cellulase enzyme for the cellulose digestion. So it was assumed for a long time that snails they digest cellulose by secreting their own cellulase. So scientists they were like of the view that snails can produce their own cellulase. But after a careful examination, it was revealed that the cellulase activity was present in the intestinal contents of the snail. It was an indication towards a major role of intestinal microorganisms in the digestion of cellulose in case of snails. So here also, first it was assumed that snails can produce their own cellulase, but after a careful examination by the scientists, it was discovered that snails are not producing cellulase of their own, but they also have got intestinal symbiotic bacteria or microorganisms which are producing cellulase for the snails. Now this example is of silverfish. So this fish is very common in our homes. It may dwell our books. So you may have seen the silverfish going here and there in your books. So the gut of this fish or the silverfish has been reported to contain a number of microorganisms, but no cellulose digesting species has been discovered. So this is a very important point, that many microorganisms they have been reported, but those microorganisms they are not functioning for the digestion of the cellulose in silverfish. So this report it proves the fact that the silverfish has its own cellulose digesting enzymes. So this is a very important point to note, that silverfishes they may digest cellulose of their own, and they are not dependent upon the microorganisms for the production of cellulase enzyme. Now silverfish can survive only on a cellulose diet. So they should feed on cellulose diet, otherwise they will not survive. Now the case of termites. So termites are known for their wood loving behavior. So they are eating wood like anything, and they are taking in lots and lots of cellulose, and they are digesting it. So now we have to see that how the termites they can digest this cellulose. So termites are well known for their wood destructive behavior. So termites they live exclusively on wood. So you may have seen that either the termites they are living in tunnels that they are making of some material, or they are living in wood. So they are not coming in the open environment. So you must not have seen the termites in the open environment. So there is a very important reason behind this behavior, that why they are living inside wood or tunnels. So I'll be taking that topic later on. So termites they are living exclusively on wood. The intestine of termites is having different kinds of bacteria and flagellates. So here you should emphasize on flagellates. So flagellates may be protozoans. They are protozoans, as you have seen, as you have studied Euglena. Euglena is a flagellate. But here, there is a colony of different types of bacteria and flagellates in the intestine of termites. So here I want to emphasize that termites are not having only and only bacteria, but they are also having flagellates. Now these flagellates, they are found from the termites. They have been found to be obligate anaerobic in nature. So this is a very important line to emphasize upon. The flagellates, which are responsible for the digestion of the cellulose in the termites, they are obligate anaerobic in nature. That means they are not facultative. So one type of nature is obligate or obligatory. Second type is facultative. So facultative is, what will be a facultative anaerobe? That animal or that species which is living in anaerobic conditions, but if it is giving oxygen, if it is given oxygen, it will shift to aerobic respiration. So that will be facultative anaerobe. But here the flagellates, they are obligatory anaerobic in nature. That means if they are given oxygen, they will not survive in that particular medium, oxygen medium. So they are obligate anaerobic in nature. So they need anaerobic environment for their living, for their survival, for their digestive action. Thus, these flagellates can only live in an oxygen-free environment. So that I have explained to you that why they need oxygen-free environment. If termites are exposed to oxygen, now this is a very important point. These symbiotic flagellates will be killed in an hour, but the termites will survive. Now I think you are getting the answer to my question that why termites are not coming to the external environments, why they are present inside the wood, why they are present in the tunnels, why they are not coming out. Because if they are exposed to oxygen, the symbiotic flagellates in the intestine of these termites will be killed in an hour. So, if they are killed, those termites, they will not be able to digest the cellulose. So, if they are not digesting the cellulose, they will again die. So, they have to remain in an anaerobic or oxygen-free environment. So these termites without symbiotic flagellates are unable to digest the wood. So that means that if they are not digesting wood, they will die. So the bacteria which are present in the termites, so what has happened that in oxygen medium, the flagellates, they will die, they will be killed, but bacteria are still present in the termites. The inefficiency of the termites to digest cellulose in the absence of flagellates, it proves that flagellates have a major role to play in the cellulose digestion and not the bacterial fauna that are present in the intestine of termites. So, termites, they rely upon flagellates more than the bacteria for cellulose digestion. So that was a very brief kind of summary for the digestion of cellulose in termites. So here I want to summarize this slide. First of all, termites, they need oxygen-free environment. Number two, they are having both bacteria and flagellates in their intestine as symbiotic microorganisms. Number three, these flagellates, they are responsible for digesting the cellulose. Number four, The termites, they do not rely upon the bacteria for the cellulose digestion. They have them, they have the bacteria, but those bacteria, they are not fully capable of digesting the cellulose. So that means they need flagellates, and for flagellates, they need an aerobic environment. If the bacteria are they were capable of the digesting the cellulose, then might be the termites may come out of the wood and they may live in the open environments. Then they would not be having any kind of problem, I think. So, because they need flagellates, they have to live inside the wood or inside the tunnels. So, students, that was the summary of termites. Now, this is about the importance of the flagellates in termites. Just see this equation or the figure. First, the termites have got bacteria and flagellates both. Next, if they are exposed to oxygen, the flagellates will die. 3. Termites, they are now having only bacteria. They are unable to digest cellulose. So that's why the flagellates, they are very important in cellulose digestion. Now, symbiotic digestion of lignocellulose in termite guts. So this is a very recent study. Now here, the ability to degrade lignocellulose gives the termites an important place in the carbon cycle. So as you know that cellulose is basically a carbohydrate, and carbohydrate has got lots and lots of carbon because carbohydrates are made up of C, H and O. So that's why the termites, they have got a very big contribution in the carbon cycle. Now this ability, it relies on their partnership with a diverse community of bacterial, archaeal and eukaryotic gut symbionts, which include the flagellates, which break down the plant fiber and ferment the products to acetate and variable amounts of methane. So that was a fermentation reaction which we had discussed in ruminants. So it produced acetate and variable amounts of methane. So with the hydrogen as a central intermediate. So that was the basic fermentation reaction. The mineralization of humus components in the guts of the soil feeding species also contributes to nitrogen cycling in the tropical soils. So this is again a very important point. In the guts of these termites, mineralization of the humus components, as they are taking in the soil also along with the woods, along with the plant materials. So mineralization of humus takes place inside the gut of these termites, and they also contribute to nitrogen cycling along with the carbon cycle. So this is the importance of termites as ecological carbon and nitrogen balancers. Lastly, the high efficiency of their minute intestinal bioreactors. So what are intestinal bioreactors? That means their intestine, as many reactions are going out in their intestine. So that's why we have here mentioned intestinal bioreactors. So that makes termites the promising models for the industrial conversion of lignocellulose into microbial products and the production of biofuels. So that is a very important aspect of termites, that we can use termites in the production of biofuels. Now this slide, it shows the formation of methane in the gut of the termites. Now, the whole gut of the termite is divided into three major parts. First is the foregut, second is the midgut, and third is the hindgut. Now, the last part of the gut, the hindgut, is responsible, or it is having the flagellates, it is having the bacterial fauna which is taking out this reaction. Now, the reaction is shown in a circle over here, that the plant fibers or the carbohydrate part of the plant fibers, it is fermented, uh, it is reacted, and acetate is formed. Hydrogen and carbon dioxide along with the methane they are produced as byproducts. So here the methane is given out, oxygen is taken in for this particular reaction. So this is the formation of methane in the gut of the termites. Now, whereas primitive termites, they digest wood with the help of cellulitic protists or protozoans, the more advanced lineages, they have an entirely prokaryotic gut microbiota. So this is a very, very important point. The primitive termites, they were using cellulitic protists for digesting the wood. But now, as the evolution has taken place, the more advanced lineages of termites, the more advanced termites, they have developed more advanced prokaryotic gut microorganisms. So they are not using the primitive cellulitic protists. Now, this major shift in the gut microbial community or the microbial fauna seems to reflect the changes in the digestive strategies and diets. So, in the evolution phenomenon, the digestive systems, they have changed. The diet has changed to some extent. The contents of the diet, they have changed. The composition of the food, that has changed. So due to these changes, their microbial community, the symbiotic bacteria, the symbiotic flagellates, their species, they have also changed according to the ingested food. So that was the overall view of the evolution among the microorganisms inside their intestines. So students, that was all about this particular topic of symbiotic nutrition and digestion. So you may get questions on this particular part, and I think you are very clear about all the aspects of symbiotic digestion. First of all, we had taken the ruminants, we had discussed the structure of different stomachs, we have discussed the different chambers of the ruminant stomach. Then we had discussed about the functions of different chambers. Then we had taken the physiology of symbiotic nutrition. Then we had taken the fermentation process which is going on in the rumen. We had discussed the cycling of urea, the recycling of the urea into the saliva. We had discussed the digestion of carbohydrate, protein and fats in the ruminant stomach. Then we had taken up the invertebrates, the cellulose digestion in the invertebrates. We had discussed many examples, including the shrimps, snails, the termites, and then we had discussed at last the physiology of the digestion, the importance of the flagellates. So, as a whole, if you take this topic as a one topic, then you may get a question of 15 marks in your final examinations. So you are supposed to give both the invertebrates and the vertebrates for symbiotic digestion. So you may give more of the examples, like you may give the example of rabbit, you may give the example of horse, so you may give more examples of invertebrates. You may give uh, the flagellates which are uh, seen, which are present in the intestine of the termites. So you may get more of the things to put in this particular topic. So that will comprise your whole of the 15 marks question. Thank you.